Hi everyone, my name is Sofia Ubieta, and I am a member of group number 6, which consists of Pedro Vera, Antiro Riley, Giovanna Morales, and Luca Cabral. Our experiment is going to be based off of biomolecular techniques. In this experiment, we aim to isolate and examine DNA from dried peas using a series of biomolecular techniques. This experiment will utilize common laboratory reagents and techniques to break open plant cells, dissolve proteins, and precipitate DNA for visualization. For step 1, we will be preparing the pea extract. First, we are measuring 10 grams of dried split peas using the digital scale. Now, we are placing the peas into the blender and we will blend them until they are fine. Now, we are transferring the ground peas into a plastic cup and adding 50 milliliters of distilled water. As soon as we finish, we will be stirring the mixture with a glass stirring rod. In step 2, we will be performing cell lysis, which is breaking open the cells. First, we begin by adding 5 milliliters of liquid detergent to the pea mixture. Now we are adding a pinch of salt from one of the salt packets to the mixture and stirring gently for 5 minutes. After these 5 minutes, we are letting the mixture sit for 10 minutes to allow the cell membranes to break down. Now that we have allowed the mixture to sit for 10 minutes, the cell membranes have broken down. We will begin step 3, which is filtration. We are placing a cheesecloth over a clean plastic cup and securing it with a rubber band. We will begin to slowly pour the mixture through the cheesecloth to filter out solid debris. This will allow us to collect the liquid filtrate in the cup. Now that we have collected the liquid filtrate in the cup, we will begin step 4, which is breaking down the proteins in a process called enzymatic digestion. In order to do this process, we will be adding 60 milligrams of papain powder to the filtered liquid. Then we will stir gently and let it sit for 5 minutes to allow the enzymes to break down the proteins. Now that the enzyme has had 5 minutes to break down the proteins, we will begin step 5, which is DNA precipitation. We are slowly pouring ice-cold isopropyl alcohol, which is about 20 milliliters, down the side of the test tube containing the pea extract. We will not be mixing this as the DNA will precipitate as a cloudy white stringy substance at the interface between the alcohol and the pea extract. We are going to hold this test tube and let it sit undisturbed for approximately 5 minutes. The test tube has sat undisturbed for 5 minutes, so we will begin step 6, which is DNA collection and observation. We are using the glass stirring rod to gently spool and twist the DNA from the alcohol layer. We are transferring the DNA onto a sheet of paper to dry for further examination. Now we can observe the DNA structure and texture. After conducting this experiment, we successfully extracted DNA from the pea cells. The DNA appeared as white stringy precipitates at the interface between the pea extract and chilled isopropyl alcohol. The strands could be spooled and lifted using a glass rod. Our results were consistent with the expected outcome, with no contamination or procedural errors. confirming the effectiveness of our step-by-step protocol. Now that we have successfully completed our experiment, we will be cleaning up. We are properly disposing of the contents in the test tube and plastic cups in accordance with local regulations. Thank you for watching our experiment on isolating and examining DNA from dried peas using a series of biomolecular techniques.